Well, so good afternoon everyone, and welcome to the fourth seminar of our Regenerative Medicine seminar series. And it's my great pleasure to welcome and introduce today's speaker, Professor Eva van Rooij. Professor van Rooij is a group leader at the Hubrecht Institute and a professor of Molecular Cardiology at the University Medical Center, both in Utrecht. And Professor van Rooij is highly regarded as an expert in the field of molecular cardiology and in identifying novel therapies for heart disease patients. Her work has been widely recognized, having received several grants, including the ERA-CVD Consolidator grant and the VICI from NWO, and she has received multiple awards from her groundbreaking research, including the Outstanding Achievement Award from the European Society of Cardiology on Basic Cardiovascular Science, amongst others. And it's a great pleasure to have you here today, and we look forward to hearing from you.
I'm messing up my mic here. Let's see. So I'm stuck here now? No, but it's gonna be okay. Yeah, I'm freed. Okay, so thank you very much, Miguel. So it's a great pleasure to be here. So I was born and raised in Eindhoven, a true Brabantse. So a big pleasure to be here, although my mom's already angry that I'm not going home for dinner tonight. But as Miguel said, I'm a molecular biologist by training, so what you'll see is a lot of molecular biology. It might be a little bit different from what you're used to, but I still think it might be an interesting topic, and I'll just show you how we're using sequencing technologies to identify new targets that might be relevant for heart disease. Would it be possible to get some water, maybe? Okay, thank you. So let me see. Yeah, so what we're studying in our lab is heart failure. And heart failure, as you probably all know, is simply the incapability of your heart to provide your body with a sufficient amount of blood to get oxygen and nutrients to your tissues. So there can be many triggers that drive heart failure. And two of the triggers that we're studying in our lab is ischemic heart disease and genetic heart disease. So where a mutation in your DNA is actually causing heart failure. So while there's many different triggers that can cause heart failure, what's really surprising to me is that the therapeutic interventions for these patients suffering from heart failure are really generic. So they're really beta blockers or ACE inhibitors. So they really don't grasp the origin that triggers heart failure in these patients. So I think that by looking at these different triggers and these different molecular pathways that are actually responsible for driving the disease, we could do a lot better. And now by all the sequencing technologies that we have available, so really looking at the detailed signaling pathways that are activated in response to these different triggers, I think we can come up with very much enhanced therapeutic interventions. So what I'll do today is show you a couple of the technologies that we have been using in our lab to really get a detailed view on what's going wrong in these patients. And I'll exemplify this by looking at the models of genetic heart disease that we've been looking at. So as I said, genetic heart failure is caused by a mutation in your DNA, and there's different types of genetic heart disease. And there's a rough division, and the division is basically dilated cardiomyopathy, where your ventricles of your heart are dilated. You have hypertrophic cardiomyopathy, so really thickening of your left ventricular wall. And then you have arrhythmogenic cardiomyopathy. And the mutations underlying these different types of genetic cardiomyopathies are often found in specific subsets of genes. So while dilated cardiomyopathy is often linked to mutations in contractile proteins, for example, arrhythmogenic cardiomyopathy is caused by mutations in genes that are linked to the desmosome. And this is something I'll be going into a little bit deeper. Thank you very much. No, it's perfect, thank you. So what is arrhythmogenic cardiomyopathy? So to go into this, I need to explain you a little bit about the desmosome. And the desmosome is a protein complex that basically connects two cardiomyocytes to each other. So it's a protein complex that's comprised of proteins such as plakoglobin or desmoplakin or plakophilin. And once you have a mutation in one of these desmosomal proteins, these cardiomyocytes basically become detached from each other. There's a lot of apoptosis of these cardiomyocytes, and there's also an unexplained infiltration of fibrofatty cells. And what it then causes in these patients is because you have these patches of fibrofatty cells in your heart muscle, you get arrhythmia, and these patients suffer from sudden cardiac death. So a couple of years ago, the group of Alexander van Oudenaarde, who is also the director of our institute, they basically developed a technology that they call TOMO sequencing. And TOMO stems from the Greek word tomos, so section. And basically what you're looking at here is a zebrafish embryo, which they unrolled. I'm not sure you can see this, but so they took a zebrafish embryo, they unrolled it, they sectioned it, barcoded each section, and then performed sequencing. So then what you get is a gene expression profile with a very high spatial resolution going from the head position of the zebrafish embryo to the tail position. And then if you look at the Z-score here at the bottom, you can basically look at the gene expression profile of specific genes. And in this particular case, they're looking at DMRT2A, and you can see that it peaks more toward the tail position in this embryo. And what you can then look for is genes that show a similar peak pattern going from the head to the tail position in this embryo. And they were able to confirm this by in situ hybridization, as you can see here. So we figured that this might be a very nice way of looking at heterogeneous gene expression profiles in our diseased cardiac tissue samples. So what Kees-Jan Bogert, a postdoc in our lab, did is he took a human explanted heart from a patient suffering from arrhythmogenic cardiomyopathy due to a mutation in PKP2. So you can see here that this mutation is causing a premature stop in this patient. So this patient basically has half of the level of PKP2, causing this patient to get arrhythmogenic cardiomyopathy. So this is a microscopic view. Then if you look at the histology of this patient, you can clearly see that this is a healthy heart. And then if you look at the signal for PKP2, you can nicely see that PKP2 here stained in magenta is indicating this desmosomal region, so really the connection between your cardiomyocyte. And in this patient, you can see that PKP2 is really distorted in these sites of active remodeling in this patient, and more in the healthy myocardium in this patient, where you still have a lot of cardiomyocytes. You can see that also PKP2 signal is really low. So this is really the reason why this patient is getting this arrhythmogenic cardiomyopathy. So what Kees then did is he took a piece of tissue going from the epicardial surface of this patient more towards the healthy region, and then he took a rim of tissue, he sectioned it, he barcoded each section, and he sequenced it. And then what you get is a gene expression profile going from the epicardium more to the healthy region in this heart. And then you get different peak patterns of genes that you might be interested in. So looking at the peak pattern for genes marking adipogenesis, we could see that markers like PLIM1 and FAP-B4, so really looking at the adipocytes, were highly enriched in the epicardial region, as you would expect. Genes linked to fibrosis were more enriched also in the epicardial region, but more spread out of the piece of tissue that he looked at. And when he looked at markers for cardiomyocyte and cardiac contractility, they picked more towards the healthy region. So this is exactly what we were expecting to see, and you can also see that there is a clear separation in the gene expression profiles if you look at the epicardial sections compared to the more myocardial sections. So the next thing Case then did is performed semi-supervised clustering. So basically looking at the gene expression profiles that he could detect in these hearts. And when he did so, he kept the physical location of the sections intact. So the 222 different sections that he obtained. And you can clearly see that he could make a rough division of 3 types of gene expression profiles that he could detect. So the first type of gene expression profile were the sections more located to the epicardial region, and then there was a big portion of sections that resembled a type of gene expression that was more spread out, the tissue that he was sequencing. And then there were also these orange types of gene expression profile sections, which were actually interspersed between the adipogenic sections and the more myocardial sections, but also here at within this myocardial region. So again, looking at the markers for gene expression, you can see that markers for adipogenesis and fat cell markers like PLIM1 was more enriched in this orange type gene expression profile, and markers for cardiomyocytes such as Desmin were more enriched in the purple type sections. So this led us to believe that the orange or the green type gene expression profile was really the marking the areas that were enriched for the fat cells, and the purple type gene expression profile was really marking the areas that were more enriched for cardiomyocytes. But of course, we were really looking at these areas where these fat cells are taking over the cardiomyocytes, so really the sites of active remodeling. So we figured that the orange type gene expression profile was actually resembling these areas where these fat cells are taking over the cardiomyocytes. To get a feel for the genes that were enriched in these areas, we then compared the orange gene expression profile towards the purple one, so the cardiomyocyte enriched sections. And we found there, so we found genes to be enriched and genes to be underexpressed in these orange sections. So, and then when we took a closer look at their functional implications, we could find genes that were enriched were related to apoptosis or myofibril assembly, and genes that were underexpressed were related to muscle filament sliding and ATP. So this again kind of forced or underscored our hypothesis that these genes might be involved in the mechanism related to the disease, so really driving this cardiomyocyte degeneration and allowing these fat cells to take over the heart muscle. Then when we took a closer look at the genes that were actually specifically enriched in these orange type sections, we found there to be ZBTB11, which is a very ill-studied transcription factor. It has not been related to heart muscle disease. But when we took a closer look at ZBTB11 and looked at where it exactly was expressed, we could see by immunohistochemistry, as you can see here, the ZBTB11 is indicated by this darker pink signal. You can see that ZBTB11 is specifically expressed in the heart muscle cells that are flanking these fatty areas in this PKP2 mutant heart. So you can see here these heart muscle cells being enriched for ZBTB11. what was also special to see when we counterstained with desmin, which is a marker for heart muscle cells, as I mentioned, we saw that the higher the level of CBTB11, the lower level of desmin. So again, triggering us to believe that there might be a correlation between CBTB11 and cardiomyocyte degeneration. So was it specific for this PKP2 patient? No, because when we looked at the hearts of other patients carrying a PKP2 mutation, we could also see that there was an enrichment for CBTB11 in these heart muscle cells. Not only was it for patients with a PKP2 mutation, but also other patients where these fibrofatty areas are part of the disease pathogenesis. We could see that CBTB11 was enriched in some of the cardiomyocytes that were bordering these fibrofatty patches. So what does it do? So to answer this question, Case generated an adenovirus expressing CBTB11 and then treated iPS cardiomyocytes with them, so human cardiomyocytes. So he was able to overexpress this transcription factor, as you can nicely see by this nuclear staining. And then when we looked at the gene expression profile or functional implications of this overexpression, you could see that CBTB11, an increase in this transcription factor, induced the enrichment for genes related to p53 signaling, which is a clear apoptotic signaler, and there was also a reduction in genes related to cardiac muscle contraction. And then in the functional assay, we could see that the cells exposed to CBTB11 had more apoptosis. So I think this is nicely showing us that this spatial sequencing is very informative and useful if you want to look at a remodeling response throughout a tissue. So the heterogeneity of your remodeling response determines the peak pattern of your genes that you might be interested in. And so here we looked at the genes that might be involved at the site of active remodeling. But you can imagine that basically you can apply this technology to any type of tissue that you might be interested in, and then just link it to the local remodeling response that you're looking at. So for us it has been very informative, and we've done it on many different explanted hearts now, but also for different disease indications. And I think it can be very useful for identifying new genes or pathways that might be relevant. So I won't go over this in detail. I mentioned most of this, but I think there are, of course, open questions, as I already discussed with some of you today. Our job never ends. So we're now looking into how a mutation in PKP2, so one of these desmosomal proteins, can actually activate a transcription factor, and how this is then linked to a loss in desmin. And that's what we're further looking into. So while we were looking into the gene expression profile in these explanted ACM hearts, Jenny, another postdoc in our lab, she actually looked at the level of these desmosomal genes. And she obtained protein from patients suffering from one of those PKP2 mutations. So it was to be expected that you're seeing a lowering in PKP2. But when she did Western blot analysis for also these other desmosomal proteins, she saw that not only PKP2 was reduced in these patient samples, but all these desmosomal proteins were lower expressed. And this was actually only detectable at the protein level, so really indicating a protein degradation effect and not a transcriptional effect. She could confirm this by staining, so you can clearly see that these signals are distorted and lower in these patient samples compared to a control explanted heart, and this is a quantification of these protein levels. So then she did a correlation analysis, not only in ACM explanted hearts, but also in patients suffering from other genetic cardiomyopathies. And she could see that there is a clear correlation between the level of PKP2 and these other desmosomal proteins. So basically showing the more PKP2 you have, the better your other desmosomal proteins do, and if that decreases, also the level of these other proteins decline. So what we're doing in our lab, and I guess that's a system that you guys are used to using here also, is of course the IPS cardiomyocytes, or the IPS lines, which in this particular case we differentiated in cardiomyocytes. So we got a line with a pathogenic PKP2 mutation. We indeed again saw the haploinsufficiency in PKP2, so half of the level of the expected level of this protein, and this was confirmed by stainings. But then also when we looked at these other desmosomal proteins here at the bottom, you can see that these were also lower compared to their isogenic control. Also in a 3D setting, we used a microtissue model from Christine Mummery's lab, where you basically introduce different cell types to mimic a piece of heart tissue. And when we compared a wild-type microtissue compared to a tissue containing a PKP2 mutation, we could also see that these tissues were more prone to show cardiac arrhythmia. Another thing that we like to do in our lab, especially now with CRISPR-Cas9 making our life a lot easier, is to introduce these known human pathogenic mutations in the mouse genome. So what you're looking at here is a known pathogenic mutation in this PKP2 gene that we've been studying in the previous slides that I showed you. And we're basically introducing a comparable mutation in the mouse genome. So this then translates into 755 del-A mutation, but basically giving the same amino acid change. Also in vivo, this mutation led to PKP2 haploinsufficiency, again showing a lowering at the protein level, but this time also the mRNA level, of course, because you're introducing a premature stop, so the mutant allele is degraded by nonsense-mediated decay. You can see here at the bottom that the signal for PKP2 is indeed a lot lower compared to the control mice. And then also, and this is of course striking, but that's already what we saw in the patient material in both the explanted hearts and in the IPS cultures, is that by introducing a single point mutation in PKP2, you can also lead to the degradation or introduce the degradation of all these other desmosomal proteins. So I think this shows us, because we're only seeing at a protein level and not at the mRNA level, that somehow PKP2 must be functioning as an anchoring protein, and a single point mutation causing haploinsufficiency makes these other desmosomal proteins basically float around, and they are then degraded in the cell because they can no longer attach to the desmosome. So in time, this also translates to cardiac dysfunction, and we get cardiac fibrosis. Somehow mice are resistant against this fibrofatty remodeling. They're not really showing this, no matter what model you're looking at. But we can still see characteristics that we can find back in these ACM patients. So, and when we perform stainings for these desmosomal proteins, as you can see here, this is a wild-type mice showing these nice desmosomal stainings of these proteins. You can see that it's lower in the mutant mice, and that it's especially distorted. We call it desmosomal disarray at these sites of active remodeling. So where there's a lot of remodeling taking place, then these signals really become more important, we think. Because we wanted to figure out what's actually the driving mechanisms in these patients, we took tissues from the mice at a time point where they aren't showing any cardiac remodeling yet. And we performed proteomics analysis, so really looking at the protein changes that are occurring in these mice, comparing wild type to PKP2 heterozygous knock-in mice. And we found, as expected of course, a lowering in the level of desmosomal proteins, but we also saw a reduction in proteins related to ACM and adherence junctions proteins. And so that we can also confirm these changes. For example, when we look at the adherence junctions proteins right here, you can see that by Western, indeed we could confirm a lowering not only in the desmosomal proteins, but also in other proteins that are relevant for the interaction between neighboring cardiomyocytes. So then Jenny, of course, wondered, okay, these adherence junctions, are they also distorted in the explanted human heart, so in the patient suffering from arrhythmogenic cardiomyopathy? And that's indeed what she saw. So also in these patients harboring a PKP2 mutation, there's a lowering in these adherence junctions proteins, and the signal seems especially distorted at these sites of active remodeling. A certain class of proteins that we actually saw induced in these mice are proteins that are related to the ubiquitin proteasome system, the UPS. So that was of course interesting because we saw degradation of a lot of proteins that might be related to the disease. So then one way to manipulate UPS is actually by using a UPS inhibitor. In this particular case, we're looking at MG132, which is a known inhibitor of UPS, so protein degradation. Of course, you cannot block protein degradation in mice forever because that will cause issues, but what Jenny did here, she injected MG132 for 10 consecutive days and then looked at the protein levels in these mouse hearts to actually confirm that UPS was at least partially responsible for the degradation that we were seeing in the desmosomal proteins. So this is a lot of information, but I want you to focus on this set of proteins and then looking at the effect of MG132 in these mice. So here you can see this. So what we're looking at here is we're seeing a slight yet significant increase in some of these desmosomal proteins in these mice after we block UPS, really showing us that it's at least partially responsible for this degradation. Of course, this also leaves us with a lot of questions, but I do think it showed us the value of using these knock-in mouse models, because there would have never been a way that we could have come up with these revelations by using the floxed allele of PKP2, for example, because the floxed allele, so genetic deletion of PKP2, that model has been around for quite some time. But apparently there's something special about introducing a pathogenic mutation in a gene and causing haploinsufficiency that way. Because in the genetic deletion, they never observed this decline in desmosomal proteins. So it's really something specific that has to do with protein stability and this mutation. So, but of course, I guess you guys also know CRISPR-Cas has really made our life a lot easier because introducing a pathogenic mutation or whatever mutation you are interested in has become way more easy than it has been. Okay, so I showed you that haploinsufficiency in this desmosomal protein we think is a key driver for the disease. So we have a mouse model or a human model, the IPS model, showing us this haploinsufficiency, but also the mouse model. This translates into a lowering of these desmosomal proteins, and this then also leads to a functional decline in these cardiomyocytes or in these heart tissues. So then Irene Kyriakopoulou, a PhD student in our lab, she figured, well, if it's haploinsufficiency that's driving the disease, why not reintroduce PKP2 to restore its physiological levels by using an AAV vector, so an adeno-associated vector, simply supplementing PKP2 back in these cardiomyocytes and seeing whether that can restore its function. So she generated a CMV-driven human version of the PKP2 gene, which she introduced in the iPS cardiomyocytes with this PKP2 mutation. As you can see here, she could restore the physiological level of PKP2 back to its level that we were seeing in healthy cardiomyocytes. But then also when she looked at additional proteins like UP and desmoplekin, also these proteins were restored. And this really surprised me a lot, because it simply means that if you reintroduce PKP2, it somehow finds the desmosome again, and then it allows these other desmosomal proteins to basically latch on and prevent them from being degraded. And that's also what she quantified. So then, of course, she also wanted to test this in vivo. She used a mouse version of PKP2. She MIG-tagged it. You can see here that she's able to introduce it in the mutant mouse heart, and we're also seeing an increase in PKP2 with a slight shift here, which is introduced by the MIG-tag. But then also when we look at the other desmosomal proteins, they are again restored. And this is also true for some of the adherence junctions proteins that are reduced in this mouse model. So I think this is really, this could be potentially really important for a lot of ACM patients, because many of these patients, and not only the ones that are harboring a mutation in PKP2 itself, but also in the other desmosomal proteins, are getting the disease because they don't have a sufficient amount of PKP2. So we teamed up with NWO Therapeutics, which is an American company, and we weren't the first to think of this approach. Of course, that's a pain, or a pity, but they are thinking about it too, and they're actually starting a clinical trial at the end of this year. And I think it can really be very important for these patients to see whether supplementing PKP2 can actually help them too. So we're teaming up with them now to submit this next month. Yeah, I think gene therapy by itself is, of course, very valuable, but I think these AAV factors are far from ideal. So any material that you guys, the tech-savvy guys, can come up with to actually bring these medicines or these gene products to the heart will be of great benefit, because AAV is not ideal as a delivery vehicle, I think. Yeah, to take a little sidestep, so using the iPS cultures, of course, a lot of us are differentiating these cells into cardiomyocytes, but another approach we took, Arwa Kohela, a PhD student in our lab, did. She actually wondered why these fat patches are appearing to be originating from the epicardial surface in these explanted hearts. So to try to pinpoint this, she actually differentiated iPS cells, wild type and ones harboring a PKP2 mutations, into epicardial cells. And you can see here by the expression of WT1, which is an epicardial marker, that they're as epicardial-like at day 30. And this is also confirmed by this Western, showing a nice signal for WT1 up until day 27, and this is the quantification here. So while they are still as epicardial-like, when we look at the RNA gene expression in these mutant epicardial cells compared to wild-type epicardial cells, so not doing anything specific to them, these cells already show an upregulation of genes that are involved in pathways like dilated cardiomyopathy, extracellular matrix, cell adhesion, which of course are key aspects of disease remodeling in these ACM patients. But we were really intrigued by this functional annotation, so adipogenesis. So if you guys know anything about epicardial cells, these cells are cells that can differentiate. They can undergo EMT, so epithelial to mesenchymal transition. They can give rise to many different cell types originating from this epicardial surface of the heart. So the fact that adipogenesis is among one of the functional indications might actually imply that they are also capable of differentiating in these adipogenic-like cells. And this is also what we could confirm when we looked at the gene expression of, for example, PPAR gamma, which is an adipogenic marker gene. You can see that it's strongly induced in the black bars, so the epicardial cells from the PKP2 mutant line compared to the control line. And there's also an induction in fibronectin, so a gene related to fibrosis. And then when we performed an oil red O staining on long-term cultures of epicardial cells with such a PKP2 mutation, you can see the presence of fat droplets in these cells. So something we like to do is we do a lot of single cell sequencing, so looking at the gene expression profile in the individual cells. In this particular case, it didn't really show us specific subsets of epicardial cells, but what it was interesting is that we could clearly separate the mutants from the wild type, or so the control epicardial cells. But then when we looked at marker genes for epicardial cells, they were only expressed in the corrected line. But then when we looked at marker genes for adipogenesis or fibrosis, they were clearly enriched in these epicardial cells from the mutant line. So showing us that a single point mutation in PKP2 makes these epicardial cells spontaneously undergo some kind of transition to make them become more fatty-like, fibrosis-like. So we think that this is a clear indication that the fibrofatty remodeling in these patients might actually be caused by these epicardial cells. So to make a long story short, we identified a transcription factor to be enriched in these epicardial cells that are starting to differentiate in these mutant cells. And when we performed an SI experiment, so knocking down this particular transcription factor that we now know to be AP2A, in these mutant epicardial cells, we can lower the expression of these fibrofatty genes. And conversely, when we only overexpress this transcription factor in healthy epicardial cells, we can induce this fibrofatty remodeling. And that's also what you see here at the bottom with this immuno. So epicardial cells become more positive for alpha smooth muscle actin. So you can spontaneously drive fibrotic phenotype. And also we can see the appearance of these fat droplets in these epicardial cells, which they normally never do. So this shows us that these epicardial cultures, and I think the IPS modeling is showing us great insights related to cardiac disease, and that's also, of course, something that you guys very well know, and you can also use it to identify transcription factors or genes or pathways that might be relevant for your disease of interest. So also when we look at the expression of AP2A, also known as TFAP2A, in these explanted ACM hearts, you can see that it's indeed enriched at the epicardial surface in these patients, and that's something that was previously not appreciated. So this might of course be therapeutically relevant to inhibit AP2A, but currently we do not really know whether inhibition of this fibrofatty remodeling is a good thing, because are you losing cardiomyocytes because of this fat layer infiltrating in? So is the fat outcompeting your muscle layer? Or are your cardiomyocytes degenerating and are these epicardial cells simply filling up the voided space? So that's something that we're looking into now, and we're further pursuing the interaction between the fat cells and the heart muscle cells and who's actually competing with whom. So I want to briefly talk about a last story, and then I'll quit. It's related to actually hypertrophic cardiac remodeling. So it's basically a disease that's often caused by a mutation in one of your sarcomeric genes, so myosin binding protein C3 or MYH7. And this causes hypertrophy, mainly of your septum wall, so the wall between your right and your left ventricle. and you can see that it's a severely remodeled heart. So we are, at our institute, we're using a single cell technology, single cell sequencing technology that it really requires fresh tissue. And of course the access to human fresh cardiac tissue is a problem. But these patients that are suffering from hypertrophic cardiomyopathy, they actually get a septal myectomy. And during this procedure, these patients basically undergo surgery by which part of their septum is removed to alleviate them from their outflow tract obstruction. So there's this thickened piece of heart muscle. It's basically causing your outflow tract to be obstructed. The septal myectomy then relieves this, and then it also provides us with a piece of human tissue, cardiac tissue. So we lyse this tissue, we put it through a fax, so a sorting machine, and then again collect individual cells, barcode them, and sequence them. So like any other single cell RNA sequencing paper, you then get different clusters of cells that you can analyze. But something that I really liked seeing in this assay is that when we looked at the individual, so when we combined all the cardiomyocyte that we got, we of course could perform correlation analysis, so looking at a key gene and then linking the expression of other genes to this. But when we looked at the correlation in the individual patient samples, like we did here in the first 5 rows, you can see that we could identify a comparable gene correlation. So this really provided me with support that we're not looking at some bioinformatic trick or something, but we're really looking at biology. So this gene correlation did occur also in the individual patient samples, and we're now looking into some of these genes to identify their relevance. This approach, it's called CNIC, actually allows you to identify regulons of genes. So genes that are regulated in a comparable manner. And because they are regulated in a comparable manner, you can also identify transcription factors that might be correlated to their expression profile. And for example, I just highlighted 4 of them here, and you can also look at this UMAP. You can see that the expression of these transcription factors is really specific for a subset of these genes. So really maybe pointing towards biology that we're looking at here. So now we're performing experiments by which we're overexpressing these specific transcription factors to see whether they can drive some of this hypertrophic remodeling. And then another aspect, because we're using flow, the sorting, to identify or to collect our single cells, you can also use cell morphological data to actually link this to gene expression profiles. So here we're using the forward scatter area. So once a cell passes through your sorter, you can basically identify its cell size. So we're using forward scatter area as a proxy for cell size. So you can really identify the larger cells and then link the gene that are expressed in these cells to them. So I just wanted to point this out there. It's a recently published story about it. I think it really shows you how you can use the facts, the data that you get from your sorting procedure and how to link that to gene expression. I won't talk about this, but I do think that this is something we'll be talking about a lot in the next 5 or 10 years. So it's gene editing. We are involved in this network, and I don't think you can talk about genetic cardiomyopathies without talking about gene editing or gene therapy. So this is of course all related to what we can do now with CRISPR-Cas or the base editing or prime editing, because we can basically revert any mutation that there is right now back to its healthy state and then maybe have an impact for these patients. But also here, delivery is really the problem that we have. So anything you can come up with to get these tools into the heart would be life-changing. So I'll stop right there. This is the team, our collaborators and our funding agencies, and I'm happy to discuss any questions that you might have. Thank you.